This lecture will focus on another medically important flagellate, which is Trichomanas vaginalis. The picture shows an image from scanning electron microscope. This is the trophozoite stage of the protozoan. We can see the flagella extended out from the main body. Okay, I invite you to scan this QR code to see how these protozoa move on the YouTube. Okay, Trichomonas vaginalis is grouped under the order of Trichomonadida. So, apart from the pathogenic species, they are also commensal Trichomonas species. For recall, so flagellates of medically important include Giardia lambda and Trichomonas vaginalis. So Trichomonas vaginalis is the protozoan that colonizes the mucous membrane of vagina. We will look into the epidemiology, morphology, life cycle, pathogenesis, clinical manifestation, and diagnosis related to this protozoan. For epidemiology, Trichomonas vaginalis infection is a sexually transmitted disease. The disease is worldwide distributed, as the life cycle can be completed only in the human host. Both male and female are susceptible to this infection. The high-risk group include those with multiple sex partners or with other sexual-related disease. The infection is higher among age group of 16 to 35 years old. It rarely happens in pubertal or post-menopause women. It might be due to hormonal factors. Okay, Trichomonas vaginalis exists only in one life form only, which is the trophozoite stage. They are usually found in the female lower genital tract or the male urethra and prostates. The shape looks like a pear, and it has 4 anterior flagella and 1 lateral flagellum attached to the body surface. So it has a exhaustal component that runs down in the middle of the body and ends in the posterior pointed end. It has a round nucleus located in the anterior position. The life cycle of this protozoan involves only the trophozoite stage. The trophozoite is the infective and also the diagnostic stage. Okay, the trophozoite is usually passed from one human to another human through sexual intercourse. Okay, the trophozoite can grow and multiply in female lower genital tract and also in the male urethra and prostates. So this trophozoite is usually multiplied by longitudinal binary fission. So when they colonize the human body, they live on the mucosa and they feed on bacteria and white blood cells. For clinical manifestation, the disease caused by Trichomonas vaginalis is known as trichomoniasis. It is a very prevalent sexually transmitted disease. So the trophozoite are usually passed from one human to another through sexual intercourse. And they usually grow and multiply in the male and female genital organs. So they feed on the mucosa and white blood cell in the localized mucosa area. In female, the infection is frequently symptomatic. It is present with vaginal discharge. The discharge could be yellowish, greenish, or presented with unpleasant smell, or frothy with bubbles, contain a large number of parasites and white blood cells, causes local irritation and burning sensation of the vulva or vagina. Urethritis and cystitis presented with dysuria, difficult to urea, or urine at the night. In pregnant lady, it causes low birth weight and preterm delivery. Therefore, it is important to screen for this disease during pregnancy. Okay. In male, the infection is frequently asymptomatic. The symptoms is usually mild if it's symptomatic. So the male will experience itchiness or discomfort inside the penile urethra. Occasionally, urethritis or epididymitis or prostatitis can occur. Okay, in terms of the diagnosis of trichomoniasis, the routine diagnostic method include microscopic examinations. So the samples could be the urine samples, vaginal secretion or scrapings, or in male, the prostatic secretion. So special stain could be done as well. The special stain include papinocolor, Giemsa, Leishman, or ashes shift stain. So to make it easy for us to visualize the trophozoites, we can also use fluorescent labeled monoclonal antibody that will bind specifically to the trophozoite. It will make the trophozoite fluorescent under the microscopes. For culture, it was done previously in maybe 1990s, but it was not done today, as the culture methods involve the culture of the clinical specimen in a special culture medium, and we have to wait for the growth of the parasite for maybe more than 5 days. Okay, so due to the tediousness and also the contamination issue of the culture, it is not done today. For serological and molecular tests, ELISA can be used to detect the antigen or the protein of the parasites. Whereas if we want to use blood tests, the antibody against the parasites can be detected in the serum sample as well. So apart from the antigen test, we can also detect the presence of the parasite DNA in the clinical samples. It's either urine or the prostatic secretions. The treatment for this infection is metronidazole or tinidazole, similar to the infection of other anaerobic protozoa like entamoeba or giardia. So all anaerobic organisms, we will usually treat them with metronidazole. However, in this case, the special thing is that we have to identify and treat all the sexual partners of the patient as well to prevent the continuous spread of this disease. Okay, these are the trophozoites of these parasites. So we have the image from saline wet mouth, Giemsa stain, ion hematoxylin stain, or fluorescent antibody staining method. For saline wet mouth, okay, this is the sample from a saline wet mouth. We can see the 4 flagella extended from the protozoan body, and in this case, the nucleus and the lateral flagellum are not visible. So for Giemsa stain, we can see the pear shape of this parasite. These are the flagella extended from the parasite body, and this is the central exostyles in the middle part of the parasites, and the black stain round component are the nucleus. Okay, these are the specimens from ion hematoxylin stain samples. In this case, we can only appreciate the pear shape of the trophozoite and the nucleus structure. The flagella or the exostyle body are not visible in this preparation. Okay, this is the fluorescent stain samples. We use antibody conjugated with the fluorescence that specifically bind these trophozoites. So this staining method is visualize the exostyle body and the flagella of this protozoa. Okay, that's all for this session. Thank you for your attention.